A very big hello to my cutest little champions out there. How are you all? Tell me in the comment section. Well, I hope that you are doing absolutely great. If at all you are not doing great, I am here. Do not worry. We'll make everything super fine. Okay. And I also hope that you have watched the one-shot lecture and all the one-marker question video. If at all you have not watched it, please watch it, because I had made sure that I explained to you each and every concept behind whatever questions were being put down there. And when you clear your concepts out there, my dear children, you will be so confident such that, okay, if that same concept comes in a 2-marker or a 5-marker, you will be able to answer it very, very easily. So that is how I try to teach you. And if at all you think that's true, what you have to do, click on the like button right now. And in today's session, we are going to cover all the 2 and 5-marker questions that comes from this chapter. Ma'am, what about the other 3-marker question and all is not there? Well, it's not there according to the blueprint that they released the previous year. So if you see, there would be 1 2-marker and 2 5-marker. LA means long answer. So 2 5-marker questions coming from this chapter. Again, 1 2-marker, again 2 5-markers, 1 2-marker and 2 5-markers. Rest all of it goes to the MCQ questions that we already discussed. 1 MCQ question will definitely come from this particular chapter as well. Okay, so are we ready to clear it up? And I'm pretty sure those of you who watched the previous video on the 1-marker question, they would be feeling that this is very, very easy. I'm definitely gonna score 13 on 13 in this particular chapter. So let's begin it with the first question that we have for a 2-marker question, that is, write a short note on sex determination method in birds. What did we discuss? How is sex determined in the case of birds, my dear children? It's very unique, because in the case of birds we have ZW, ZZ type of sex determination. For the first time, it is the females who are going to give rise to 2 type of gametes. Usually we have seen it's in the case of human beings, the female produce 2 type of gametes, but both of them same only. Have X chromosome here, have X chromosome here also. XX is her genotype. For example, if I am a female, I would be having a genotype that is what? XX. In this case, the females have ZW genotype. That means the gametes that she is going to produce would be what? Either would be Z or it would be W, my dear children. So here 2 type of gametes are being produced, and what do you call this? This is known as female, since it is happening in female, heterogamety. Heterogamety means 2 different type. Hetero means other, different. 2 different type of gametes are being produced. But in the case of males, if you see, they produce just 1 type of gamete. All of them have what? Z chromosome itself. Now let's see a crossing and see how it happens actually. So over here I have ZW, over here I have ZZ. The gametes would be Z and W over here, Z and Z. In this case, this and this crosses, you get what? ZZ. Tell me it's a male or a female in the comment section below. ZZ is a male or a female? It would be what? A male, my dear children. Right? So this is a male. Now this cross with this, again you get ZZ. Again, what do you get? You're getting male itself. 2 males. Okay, now it's time for females. ZW, you get females. Okay, and ZW, again you get female. So 50% chance for a female to be born, 50% chance for a male to be born. This is the sex determination method in the case of birds. And since the female is the one that produces 2 type of gametes, you call this female heterogamety. Can we see the answer? How do you frame the answer? So 2 different types of gametes in terms of sex chromosomes are produced by females. That is known as female heterogamety. Give a small introduction. Then say the female produced 2 type of X, one carrying Z, another carrying W chromosome. Whereas the males, they produce only 1 type of sperm carrying what? Z chromosome itself. So if now you gave the understanding of the meaning of what you mean by female heterogamety. Okay, and you also said that in males this is not happening. Now you say, if the sperm carrying Z chromosome fertilized with the egg carrying Z chromosome of the zygote. Okay, egg is carrying Z chromosome and the sperm is carrying Z chromosome. What will happen? The zygote will be developing into male. Why? Because the genotype of the zygote is what? ZZ. You can explain it better, lot better. Okay. Now, if the sperm carrying Z chromosome fertilized with the egg carrying what? W chromosome, what will happen? The zygote will produce into a female. Over here, ZW is this thing. And make sure you are making this particular thing while you're answering, because sometimes they do not see the whole answer like this. So boring for them also, no, for the teachers. So let's make it easy for them. Okay, you can write it properly. If at all not this seems to be very complicated, you can just go ahead with this particular thing. This is totally enough. Okay, this would be totally enough. And whenever you write like this, please make sure you're writing parents over here, and then you're making sure it's gametes, and then you are saying it's F1 generation. So parents, gamete, F1 generation, line by line you had to explain. Okay, and in that case you get the 2 marks completely. Now going ahead with the next thing that we have is what? Question number 2. What is haplo-diploid sex determination mechanism? Mention an animal which exhibits this. What do you mean by that? What do you mean by haplo-diploid sex determination? So in haplo-diploid sex determination, it says that the haploid, suppose the number of chromosome is half, which you call as N. Okay, suppose someone has 32 number of chromosomes, but this organism only has 16 number of chromosome. Don't you think it's the half of it? Yes, it's the half of it. So I'll call this as this is haploid in nature. Now when an organism have haploid or half the number of chromosome, it becomes male. Now on the other hand, if it has double the number of chromosome, double means 2n, I can write half was n, double will be what? 2n. So over here, when it is 2n, it would be a female. Didn't understand? I'll give you an example of it. Here also an example is asked. Mention an animal which exhibits this. What is that animal, my dear children? It is honeybee. Honeybee shows this. So over here, in the case of females, they would be having, the females are always diploid in nature, 2n. Okay, if I talk about the males, they are haploid in nature. That means they have n number of chromosome. 2n means in the case of honeybees, it's 32 chromosome, and half n means it is 16 chromosome out there. Now, this female wants to make gametes. Okay, so what will she undergo? Usually what do we undergo? Whenever we want to make gametes, we undergo some process which is known as meiosis, reductional division. So that in the gametes, I get half of the chromosome only, because I am expecting the other half will be coming from the male partner, which is why I will be having half here, half here. Okay, so this particular female underwent meiosis, got the gametes which were haploid in nature. Now what about the male? Male is already half. Can it go again undergo reductional division? No. It says I don't want to undergo reductional division. Instead, I will undergo equational division. Whatever number of chromosomes over here, let me make the same number of chromosomes over here. For that, it has to undergo mitosis, not meiosis. Now, my dear children, what will happen? If I want to make a female honeybee, what will I do? This particular gamete from the female should fuse with another gamete from the male. Right, that's when you get n and n becomes what? 2n. That means this would be female. Ma'am, then when will male be produced? The male will be produced when this particular gamete which is produced by the female is not at all fertilized. Okay, when it is not fertilized, it will automatically form into what? The male organism out there. So this process has a name. Whenever without fertilization, what are happening? You're forming an organism or a male organism. You call this as parthenogenesis. Okay, parthenogenesis, which is very unique in the case of honeybees out there. Okay, so when n and n comes together, you get female. When n is not fertilized, you get male. And the males are generally just for sexual activity. They are known as drones. Okay, they are known as drones, and they are just for sexual activity. In the honeybee society, there is only one queen bee available. Okay, this queen bee goes for a nuptial flight, as in a flight where she undergoes sexual intercourse. So there might be so many drones coming up and trying to, have a relationship with it, but only the fittest one could be surviving. And after the sexual activity is being done, this drone gets killed as well. It dies off. Okay, so it's a very different story that they have, but this is how sex determination works in the case of honeybees. So haplo-diploid sex determination is this, where diploid will be always female and haploid will be always male. I hope you got it. So how do you write the answer? So you can make the diagram exactly like this, or you can make how I showed you, either way. Now the haplo-diploid sex determination mechanism is a system in which sex of an individual is determined by the number of chromosome sets it inherits. Okay, what do you mean by that? It is determined by the number of chromosome. In this mechanism, males develop from unfertilized eggs and are haploid, having a single set of chromosome. Now females develop from fertilized eggs and are diploid, having 2 sets of chromosome, which is what we said, that it is totally determined by the number of chromosome sets it inherits. Okay, now Q and B lays eggs. If the egg remains unfertilized, it develops into a male drone. If the egg is fertilized, it develops into a female. Okay, it can be worker female or it can be a queen bee. Queen bee is generally not formed. For queen bee, you have to give the royal jelly. Lot of things are being there. So usually it is worker bee, and worker bee is who? The one who takes care of the whole, what do you call, honeycomb, who brings in lot of things, who takes care of all the things that is happening in there. Depending on the environmental and dietary factors, it can be either a worker or a queen bee. So queen bee is usually fitted a royal jelly, that's how she becomes queen. And there is a lot more story into it which we will not be discussing as of now. So I hope this is clear. That the question can be reframed in a way that is, please explain the sex determination mechanism in the case of honeybees. It's the same. What do you call the sex determination mechanism? Haplo-diploid sex determination mechanism. Done. Clear? Sorted? If it's clear, what you have to do, as I said, click on the like button, and if at all you say, ma'am, I have already clicked on the like button, you can comment down, you can share it with your friends who are in need as well. And now, write the genotype of the parents when their children are with A, B, AB or O blood group. So over here I am seeing that all the blood groups are there. A is there, B is there, AB is there, O is there. So in this condition, what would be the genotype of the parents, my dear children, if asked? So always understand, for a person to have A blood group, they can have a condition like this, where both the alleles are same. They can also have a condition like this, where one is O and one is A, where A is dominant, and which is why it will be A blood group itself. Okay, so over here I see A blood group is there. So A blood group probably can be this or this. Parent can be this or this. Now if you're talking about B blood group, it can be IB, or again it can be IB, IO. B is also dominant over O. Again, if you want B blood group, the parents can be this or this as well. But we have to make sure we need children of all blood group types. Okay, we need children of all blood group types. Now if you talk about AB, for AB blood group, one should be A and other should be B. Okay, over here both are co-dominant. They would be co-dominant. Now if you talk about O blood group, my dear children, you have to make sure the condition is like this. You also write like this or you also write like this. Either way it's the same thing. IO. There should not be any other counterpartner out there. That means we got to understand, for a O blood group, we definitely need IO for sure. And 2 IO should be coming. So for 2 IO should be coming, can I already write over here IO and IO. Now what all do you need? I need A blood group, I need B blood group also, and AB blood group also. So probably if something it's like this, it's IA over here and IB over here, don't you think everything can be possible? Let's see. This and this comes together, I get IA, IB, that means a person has AB blood group, co-dominant, right? Now this and this comes together, I get IA, IO, that means it is A blood group because A is dominant over O. Suppose a person has this, that means IB, IO, again B is dominant over O, so this will be B blood group. Now for IO, I just need this. So what is the genotype of the parents? It can be IAIO and IBIO. This is the genotype of the parents. I hope this is super clear. The genotype of the parents blood group is either IA, I is the, actually it means the same thing only, I means IO only, or it can be this. Now they have written in both the format. Okay, so parents would be IAIO and IBIO, then only you can get all the genotypes up over here, and I have already explained you how does this work. Okay, how do you get the blood groups? For A blood group you would be needing, wait, for an A blood group, come on, A blood group you need both IA, or else you need what? One IA and IO. In that case also you get A blood group. When do you get AB blood group? When it is IA, IB from one parent, or this can be ulta also. Here IB and that IA. Same way, you will get AB blood group. So you need to know this chart. If at all you are messing around this chart, probably you might lose your marks. Cool? Done? Fine. Now, going ahead with the next thing. Write 2 symptoms of Down syndrome and write the chromosome number in Down syndrome. First of all, what is Down syndrome? Down syndrome is a disease which comes under chromosomal disorders. That means the number of chromosomes is altered here and there. What is the usual number of chromosomes that we have? We have 46 number of chromosomes, my dear children. Out of this 46 number of chromosome, let me tell you, 44 of them are autosomes. They do not have anything to do with the sex of an organism. And 45th and 46th chromosomes are what? The sex chromosomes, or you also call them as allosomes. Either way. Okay, you also call them as allosomes. Now over here in the case of Down syndrome, a person instead of having 44 chromosomes, they have 45 chromosomes, as in 45 autosomes are there. One extra chromosome is there. Which one, ma'am? So if you talk about, this is a number as in a whole I was talking about. If you talk about the pair of chromosome, I can say there are 23 pair of chromosome. Okay, 22 of them are autosomes and 23rd pair would be what? The sex chromosome. When I talk about numbers, it will be like this. When I talk about pair, it will be like this. So 22 of them are autosomes and 23 of them are sex chromosome. Now assume the 21st chromosome which is there, you should get one from one parent, this from parent 1, this from parent 2. But suppose instead of getting a pair, you are getting one more chromosome, extra chromosome from either of the parent. So what will happen? You have 3 chromosomes now. This condition is known as trisomy. And trisomy of which chromosome? The 21st chromosome. Which is why what will happen? Obviously, the chromosome contains DNA have genes control your characteristics. So obviously, there will be certain fluctuation in the characteristics. And when this happens, when this condition happens, you get a syndrome which is known as Down syndrome. Okay, or disorder which is known as Down syndrome. And it has clearly something to do with the chromosome number. So how do you write it? Over here, what is the symptoms of Down syndrome? Individual would be short statured, the height would be very short, small round head would be there. Okay, they have furrowed tongue usually, the tongue looks like almost like this. Okay, not exactly, but yeah. And partially opened mouth will be there. Their palm will be broad with characteristics, palm crease will be there. You can clearly see the line like that. And physical psychomotors and mental development would be retarded. The chromosome number in Down syndrome is trisomy of 21st chromosome. So over here ends your symptoms. 4 symptoms. Short statured, small round head, furrowed tongue, partially open mouth. Okay. Palm is what? Broad with characteristics, palm crease, and physical psychomotors and mental development is quietly retarded. Now, the chromosome number in Down syndrome, if you say, is what? Trisomy of chromosome number in Down syndrome is trisomy of chromosome 21st, or you can just directly say they have a trisomy condition of chromosome number 21. Now the karyotype means the number of the chromosomes which are being aligned, things like that. The karyotype of Down syndrome individual is 45 autosomes. Usually we have what? 44 autosomes, and then we have either XX or XY. Over here one chromosome would be extra, so 45 A plus XY, that means it is what? It would be a male. XY means it would be a male child who has Down syndrome. If it is 45A plus XX, it will be what? It's a female child who has Down syndrome. I hope you got it. Okay, so this is the karyotype. Works? Cool. Now XY, so this is the 45th chromosome, 46, 47 chromosome there in total. Usually we have what? 46 chromosomes. Now going ahead with the next question, define point mutation, give an example of point mutation. Point mutation is where you have a problem, a fluctuation at one particular point itself, not elsewhere. Okay, one particular point. So you might have studied about sickle cell anemia, where what is happening, my dear children? You had a codon GAG. When you talk about the codons, okay, each codon which is there, these set of codes which is there, it codes for an amino acid. Similarly, you will be having multiple codons over here. So many amino acids would be coming together to forming what? To form a protein out there. Now over here there is a fluctuation, a point mutation. Point mutation means at one nucleotide base, there is a mutation out there. So instead of having A over here, okay, and let me tell you what amino acid was it coding before. It was coding glutamic acid before. This is an amino acid known as glutamic acid before. Okay, now this has changed to what? Instead of GAG, now it is GUG. So there is a point mutation that have taken up over here. Now what happens when it is GUG? It will code for some another amino acid which is known as valine. So instead of glutamic acid, you are getting a valine over here, and rest of the amino acid will be same. But still, do you think the protein will be same? No, the protein will have definitely some problems out there. It will be a whole different protein out there. And when does this happen? This is happening in sickle cell anemia, where the you know, hemoglobin is formed of beta globin chain and alpha globin chain. So the beta globin chain gene is getting mutated like this, which is why what will happen, a proper beta globin chain is not being formed. There will be some structural deformality, and which is why the hemoglobin is there in the RBC. So if the RBC contains this deformed beta globin containing hemoglobin, my dear children, what will happen? Obviously this RBC will also have some change in the shape. So because of the change in the structure, my dear children, does not have good oxygen carrying capacity, which is why the cell of the shape of the RBC would be turning into what? Sickle shape. And always remember, this is the 6th codon. This is the 6th codon where this point mutation is happening. Okay, and always remember, A comes before U, so it was A before and then it becomes U. Similarly, glutamic acid, G comes before V, so it was G first and then comes valine. I hope that's also clear. So a change in a single base pair of DNA is known as point mutation. A classical example of such a mutation is sickle cell anemia, where the codon GAG, the second nucleotide, that is A, is replaced by U, hence making the codon GUG. Note that GAG codes for glutamic acid, whereas GUG codes for what? Valine over here, which is why it will cause structural changes. Structural changes in what? In the beta globin chain of hemoglobin. Cool? Works? Okay. Now going ahead, what are the conclusions drawn by T.H. Morgan from crossing experiments in Drosophila with respect to linkage? What did he find out? T.H. Morgan, what did he find out? He found out two things basically over here, my dear children, if you see. So suppose I have a gene like this. Okay, two conditions. Just give me a second. Okay, now my dear children, so when the chromosomes lie close to each other, you have a process which is known as crossing over that happens. They cross over each other and they tend to change some materials out there. So when crossing over happens over here, my dear children, what is the thing? Suppose this part goes there and this part comes over here. Still capital A and capital B gene will be together. Still small a, small b gene will also be together. So if you talk about newer combinations which is formed, do you think there is many? No. Over here, so I will say the recombination rate. The amount of recombination is less. Why? Because the genes over here are what? Tightly linked. They are very close to each other. They are tightly linked. What I mean by tightly linked, that means the distance between them is very less. Okay. Now on the other hand, if something is happening over here, this part is going here and this part is going here. So let's see what is the new thing that we get. So capital A, which was once with capital B, will now be with small b. Similarly, small a, which was once with small b, now will be with capital B. Now you tell me, is the recombination more? Yes, ma'am. Why, ma'am? Because the genes which are there, they are not tightly linked. They are what? Loosely linked. They have so much distance between them. So this was the conclusion drawn by T.H. Morgan experiment. He experimented, he found that certain characteristics, okay, they show high amount of recombination, whereas certain other things, they do not show much high amount of recombination. It would be the same throughout generation after generation. Okay, so he was the person who gave down two terms, linkage and recombination, and his student was there, whose name was Alfred Sturtevant. I discussed with you in the 1 marker question session, and he was the one who said that, okay, now I know that, okay, recombination is high, that means they are far away from each other. And with this information, I can get to understand that this gene might be over here and this gene might be over here. So this is known as gene mapping. He was able to map the genes. Who was he? He was Alfred Sturtevant. Okay, so the higher the linkage, lesser is the recombination, and the lesser the linkage, the higher is the recombination in this particular case. That is what he concluded. In other words, genes which are tightly linked will show lesser recombination, yes, because they are tightly linked, and the genes which are loosely linked will show higher recombination. It's easy. If you have two friends who are very close, will you allow a new friend to come in? No. So tightly linked, so less recombination. If at all you have two friends but you are not that close, my dear, someone else can also come in over here. That means more recombination. Now, what is test cross? Write its significance. Test cross is basically done when, suppose I have a plant which looks tall. and I do not know whether it's tall like this or it is tall like this. So I want to find that out. How do I find that out? I do a test cross. In test cross, I am going to cross it with a recessive parent. Recessive parent, which would be having small t, small t. Recessive parent has small t only, no? So over here, let's see. What's the difference, ma'am? What will I get? So over here I have small t, and over here suppose I have capital T, capital T. This and this uses I get capital T, small t, here also capital T, small t, here also capital T, small t, and here also capital T, small t. This and this capital T, small t, this and this capital T, small t. All of them, if I see, they have capital T over here, which is why all of them would be tall. Okay, now on the other hand, the cross is like this. Capital T, small t, and small t, small t. This and this goes, you get capital T, small t, this and this, capital T, small t, this and this, small t, this and this, sorry, this and this, you get small t, this and this also you get small t, small t. Now you tell me, here two of them will be tall, but two of them would be dwarf. So when I get this condition, okay, when I get this ratio that is 2:2, that means 1:1, my dear children, I get to understand that the plant which was looking tall, it's not homozygous tall, it is heterozygous tall. Okay, and now when I get a condition where all are tall, my dear children, where the ratio is just 1, I'll conclude that okay, it was homozygous tall. This is what you mean by the test cross, where you're crossing with a recessive parent. Fine. So how do you write it? Let's see. So answer, what is test cross? Test cross is the cross between an individual showing a dominant trait. It is cross between an individual that is showing what? A dominant trait, but with unknown genotype. We are not aware of the genotype. And what? I am crossing it with homozygous recessive individual. Significance of test cross helps determine whether the dominant individual is homozygous or heterozygous. Now you do this and you get to find that out. I explained with capital T, here some other explanation is given. You can refer to this also if at all you need. So the PDF will be shared to you, you can see all of it. Okay, perfect. Okay. Now also remember, before I go ahead with the 5 marker questions, always remember test cross is an example of back cross. Ma'am, what? So back cross is done when you go back to the parent generation and you cross with it. So this is here also the same thing is happening. In test cross you are going back and doing the crossing, right? But you are specifically crossing with a recessive parent, which is why it has a specific name, that is test cross. If you're just saying you want to cross with any of the parents, you will rather refer to it as back cross. You're going back and crossing with it. Cool? Clear? 2 marker questions clear? So these are the questions that have come the previous years like monotonously. So you can expect somewhere around this. Sex determination in organism is something that is usually being asked for a 2 marker question. And 1 marker also I explained it to you, different type of sex determination. So go be thorough with it. As I said, if at all you watch my videos, all the videos, I have made pretty sure that okay, all the concepts are here and there covered. So make sure you're watching it. Now 5 marker question, the first 5 marker question is, what is phenylketonuria? Write the cause and symptoms of phenylketonuria. What is it? It is actually an autosomal recessive disorder. What is it? It is autosomal recessive disorder. Ma'am, I don't understand it. So that means the genes of this particular, what do you call, the genes that controls this particular thing is located on what? The autosomes, not on the sex chromosomes. So that is why it is autosome. Now why it is recessive? So my dear children, when I get 2 chromosomes that have the affected gene, that's only when I can get a disease. It can only show up in a recessive condition, homozygous recessive condition. So I should get 1 chromosome from one parent, another chromosome from another parent, both of them should be defected. That's when I'll get this disease. Otherwise, I'll be just a carrier of this. I would not get this disease. That is what I mean by recessive disorder. It cannot be expressed. It cannot express itself when one is normal and the other one is defected. Because one is normal, so it's overshadows the effect. Now it's on the autosomal chromosome, which is why it is known as autosomal recessive disorder. I hope that's clear. Now, what exactly happens over here? So a gene that codes for a very important enzyme which is known as phenylalanine hydroxylase. Phenylalanine hydroxylase is affected. Now this particular enzyme, is it being formed? No. What does this actually do? I am going to tell you that. So it actually converts an amino acid whose name is phenylalanine to what? To tyrosine. To tyrosine. Who does that? This particular enzyme. But is this enzyme being formed? No. So this conversion happens? No. So then what happens? This phenylalanine is going to get accumulated in our body in the form of what? Phenyl pyruvic acid. Now ma'am, is that a problem? Yes. Why? Because the accumulation of this can cause mental retardation. It can cause mental retardation. So ma'am, that's all? No other symptoms that you can see? Yes, you can see one more symptom. That is, since this conversion is not happening, tyrosine is not being formed, tyrosine is also an amino acid. So one amino acid to another amino acid conversion is taking place over here in general condition. But since this is not happening, tyrosine is not going to be formed. And if tyrosine is not going to be formed, you need a very important pigment in your body, that is melanin, my dear children. That is also not going to be formed because tyrosine is the one that would be helping in the formation of melanin. And if melanin is not being formed, my dear children, our pigmentation will go reduced. So we will have hypopigmentation, reduced pigmentation. It will be totally extra fair. Okay, it will be so fair, your hair will also not have much color out there. So hypopigmentation in what? In your skin and your hair could be noticed out there. And not just that, this phenyl pyruvic acid, okay, it is usually excreted through our, it cannot be reabsorbed in our body by the kidneys. So it is excreted through our urine, excreted through our urine. And this, since this is there, this phenyl pyruvic acid is there in our urine, my dear, what will happen? It gives a very mousy smell, a bad smell out there in the urine. A mousy smell is also there. So this particular disorder, you have mental retardation. Since excretion of this particular thing is not it's happening well, it's not reabsorbed by body, which is why what will happen, there will be a mousy smell in our urine also. And the third part of the story is what? Melanin is not being produced, which is why we will be having hypopigmentation or reduced skin and hair pigmentation. So my dear children, one gene, multiple problems out there. This is phenylketonuria, and this is what? An autosomal recessive disorder, and it is also an example of one very important thing which you call as pleiotropy. I had to explain this in the next question, but anyway, I am telling you here itself. What do you mean by pleiotropy? Where one gene is responsible for multiple character. Usually we have seen in Mendel's experiment, one gene, one character. That is a usual thing, no? So capital T for tallness. It does not have any other features out there. But over here, one gene has multiple factors. One is what? This particular thing. And the second thing, it is also reducing what? The pigmentation out there. So one gene, multiple effects. Example is this phenylketonuria, and this particular term is known as pleiotropy. Okay, where a gene codes for multiple character. Do not get confused between polygeny and pleiotropy. Pleiotropy means one gene, multiple character. Polygyny means multiple genes are there, one character. We'll come to that. So this is phenylketonuria. What is phenylketonuria? I hope you got it. Let's see the explanation of it. It is an autosomal recessive disorder. It is an inborn error of metabolism. That means you get it by birth. Okay, it's genetic. Now the affected individual lacks an enzyme phenylalanine hydroxylase that converts the essential amino acid phenylalanine, a substance found in our food, into what? Tyrosine. As a result of this, phenylalanine is getting accumulated and converted to phenyl pyruvic acid and other derivatives out there. Symptoms: accumulation of these in the brain results in what? Mental retardation. Phenyl pyruvic acid and other derivatives are also excreted through the urine because of its pure absorption by the kidney, which is why it gives a mousy smell. Infants have lighter skin, hair and eyes than their siblings without this disease. Okay, so ones that do not have this disease have a proper color of skin and hair, but over here infants who have this, okay, this disease would be having lighter skin and hair color as well as your eye pigmented color. Okay, done, clear. Okay, if at all this question comes anyhow, will you be able to answer it? I hope you do. Now question number 2 coming under the 5 marker session is what? T.H. Morgan selected the fruit fly for his genetical experiment. Give, first of all, why, and give 3 reasons. Okay, what are the 3 reasons? Why did T.H. Morgan, select fruit flies for his experiment? First of all, what was he trying to prove? He was studying the chromosomal theory of inheritance. He was trying to prove chromosomal theory of inheritance. Okay, now why did he choose fruit fly? What do you call fruit fly? You call it as Drosophila melanogaster. So there are reasons. First of all, the reason is that they can be grown really fast. So they can be grown on a simple synthetic medium. Yeah, that is also there. It do not need hi-fi things to be grown. They just can be grown with simple synthetic media out there, and they can complete their life cycle in about 2 weeks. This is very important. Why? Because if at all I want to see some changes in the organism, I can see within a span of 2 weeks. I do not have to wait for eternity for that. Now, a single mating could produce a large number of progeny flies. Single mating is there, you can get so many progeny flies out there. So I can do much more experiment. There was a clear differentiation of sexes. The male and the female flies are easily distinguishable. For your understanding, the males are smaller, okay, and the females are bigger in size. Males are smaller, females are bigger. And they have so many other features as well. So these are the 3 things that you can write. They can be grown on simple medium, they can complete their life cycle within 2 weeks, single mating can produce multiple progenies out there. There was a clear differentiation of sexes as well. Now comes to the second part of the question, that is, what are chromosomal disorders? Give an example. Chromosomal disorders, I already told you. Down syndrome, okay, where there is an addition of chromosome. Now suppose there is what do you call subtraction of a chromosome. One chromosome is not there. Then also it's a chromosomal disorder. Or suppose there is a structural deformity in the chromosome out there, then also it's a chromosomal disorders. So the disorders in human beings are caused due to absence or excess or abnormal arrangement of one or more chromosomes are known as chromosomal disorders. Example, Down syndrome, where what is happening? Trisomy of 21st chromosome is happening. Turner syndrome, monosomy is happening. So instead of a person to have XX chromosome, so from one parent you are getting X, but from another parent you are not getting X. You are not getting anything. In the gamete there is no sex chromosome. This is known as XO condition. So over here one chromosome is missing. That is why it is known as monosomy. Okay, one chromosome is only there. Monosomy of what? The X chromosome out there. This is happening in the Turner syndrome. Now Klinefelter syndrome, what is happening up there? In Klinefelter syndrome, my dear children, instead of a person to have XY, one from the mother X chromosome, from the father you should get the Y chromosome, but over here you are getting an additional X chromosome out there. Again, this is an example of what? Trisomy condition. You are getting an extra chromosome out there. Okay, you are getting an extra chromosome out there, which is why the whole thing changes. So all of their addition, deletion somewhere is happening, and these are examples of chromosomal disorders. Now, few autosome linked, this is very super important. Mostly this question is being asked. Okay, few autosome linked recessive gene blood disorders occur in human population. Among them, some are related to qualitative and quantitative problems of synthesizing blood proteins. Explain. Didn't understand anything? Let me tell you. So they are just telling you that there are few autosome linked recessive gene disorders which are related to blood. Somewhere the blood proteins are not made enough, or probably somewhere the blood proteins, the quality of it is a problem. So one is quantitative, it's not produced enough, and one is qualitative disorder. But both of them are what? Autosome linked recessive gene disorders. Example, how can you explain them? You need to know the example. So for qualitative, where the quality is hampered, the example is sickle cell anemia. Okay, when you talk about quantitative, the quantity of it is hampered. This happens in thalassemia. Okay, sorry, the spelling of thalassemia is wrong. Okay, this happens in thalassemia, where the quantity is not being produced. If at all you are not understanding it, I'll explain you, don't worry. But before that, let me tell you, both of them are blood disorders, and they are autosomal recessive disorders. That means both the genes should be defective, then only the person would be showing the disease, otherwise they would be a carrier. Now ma'am, is there a blood disorder which is not autosomal recessive but is sex linked? Yes, and that is hemophilia. So hemophilia is sex linked gene disorder, blood disorder, and these two are the only ones which are autosomal recessive blood disorders. Please remember that. And now going ahead with what we had to explain. So, the problems can be categorized as qualitative when it affects the structure or function of the protein. Quality, the structure is getting affected, which is why the function is also getting affected. Or quantitative, that affects the amount of protein which are being produced. So in sickle cell anemia, what happens is that it is a qualitative defect. Why do I call it qualitative defect? Because my dear children, there is chromosome number 11. On the chromosome number 11, you have a gene which, the HBB gene, okay, the HBB gene which codes for the beta globin chain of hemoglobin. Beta globin chain of hemoglobin. Now over here, it can be HBA and HBA. That means it's normal. The gene, the two alleles of this gene are totally normal. There's no problem. There's no need to worry. But suppose over here, there is some mutation happening in this particular allele, which is why I'll call this now HBS. This allele is muted. Now, is there a problem, ma'am? No. It is an autosomal recessive gene disorder, I told you. So over here still, since one is affected, the other one is normal, this will overshadow the effect, and this particular person would be just a carrier. Okay, but some deformity will definitely be there in the body, but she will be just a carrier, he or she will be just a carrier, they won't have the disease. But suppose over here, both the alleles are affected. HBS condition is there. What will happen over here? The beta globin chain is not going to be structurally proper. There will be some structural deformality. Structural deformality in what? In the beta globin chain. I'm not saying the quantity is reduced. The quantity still will be produced. Beta globin chain will definitely be produced, but the structure of it will have some problems out there. So it is caused by mutation in the HBB gene. Okay, glutamic acid is substituted by valine at the 6th position of beta chain, produces HBS. Okay, that is the abnormal hemoglobin instead of what? HBA. So instead of HBA, it is producing HBS. Now HBS, when both HBS are there, homozygous condition is there, they would be diseased. This RPC which is there, it would be sickle shape. Why? Because the hemoglobin is formed of a bad beta globin chain. It's not structurally proper, which is why the structure of RBC also get reduced. Because, my dear children, there is poor oxygen absorbability. The absorption of oxygen is greatly reduced, which is why the shape of the RBC gets really bad. So poor oxygen transport is going to be noticed over here. On the other hand, if it is HBA, HBS, heterozygous condition, then they would be carrier. Sickle cell trait would be there. Defect is in the quality of the hemoglobin. This is what you have to note in sickle cell anemia. Now on the other hand, if we talk about thalassemia, it is a quantitative defect. What do you mean by that? So over here I have a picture. So there is chromosome number 16 and there is chromosome number 11. I told you on the chromosome number 11 you have HBB gene. Over here one allele, over here another allele. Now suppose this particular allele is mutated in some way such that it is not working. Okay, this is not working, it does not produce enough amount of things that it has to produce. It is totally relying on this. And over here the beta globin chain will be produced, but the amount of it will be what? Less. It will be less, which is why it will be a big problem. Similarly over here, if we talk about the alpha globin chain, the alpha globin chain has two genes out there, HbA1 gene and HbA2 gene. This gene has two alleles, this gene also has two alleles. Suppose this allele is defected, only this allele. Amount of alpha thalassemia will be reduced, but not too much. Suppose this and this gene both are not there, both are defected, then it will be greatly reduced. Suppose all the 4 genes are not being produced, all the genes are not working properly or they are not producing things. What will happen? Alpha globin chain is not going to be produced at all. Here you are talking about the quantity, not the quality. Quantity of it is getting reduced, which is why you have problem. That happens in thalassemia. Always remember, alpha globin chain is on the 16th chromosome, whereas beta globin chain is on the 11th chromosome. How do I remember it? I'll tell you one very simple fund. I do not know if it is going to be helpful for you, but 6 always look like B, right? The half of B. 6 looks like the half of B. If I make like this, it will be B. But over here, 16th chromosome is not for beta, it's for alpha. It would be for alpha. And for beta, you would be having the 11th chromosome. I know it's silly, but sometimes it helps when you have so many things to study. Yes. So it is caused by mutations in HbA1 and HbA2 alpha chain or HbB, that is beta chain genes. Hemoglobin formed is normal in quality but reduced in what? Quantity. Quality is okay, but quantity is reduced. Leads to anemia, weakness and fatigue. These are the symptoms of thalassemia. Severity depends on whether one or both alleles of the defective genes are affected. I showed you there are 2 alleles for a gene. So either one can be affected or both can be affected. If both are affected, then it's not being produced at all. Get it? Defect is in the quantity of the hemoglobin produced, not the quality. So that is the example. I hope it's clear. Now, next one. Where is it? Give reasons. Give reasons for the following. A simple cut results in non-stop bleeding in hemophilia affected individuals. Hemophilia is what? Sex-linked recessive gene disorder. And over here, why does simple cut results in non-stop bleeding, my dear children? Because in their body, the genes which are responsible for production of clotting enzyme, that is being mutated, which is why clotting enzyme is not there. And if clotting enzymes or proteins are not there, what will happen, my dear children? Will clot happen? No, which is why there will be bleeding, 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 bleeding. Turner syndrome affected females are usually sterile. Why? Because ma'am, instead of having XX over here, they do not have another chromosome, it's XO, which is why the ovaries are rudimentary, they are not fully developed, which is why do you think they will produce X out there? No, they are sterile. Okay, they are not fertile, they are sterile, they cannot produce X. Now in Morgan's dihybrid cross experiment on Drosophila showed that flies having genes for yellow body and white eyes exhibited less recombination. So you don't need to be concerned about the characteristics. If at all this term is there, less recombination. If they are showing less recombination, new recombination is not happening, right? That means what? These genes which you are talking about, they are very close to each other. The gene coding for yellow body and the white eyes, they are very close to each other. That is what you mean. Now, inheritance of skin color in the humans show different phenotypes. Now, this is an example of polygyny. It is saying, when you talk about the skin color, we all have different physical appearance, phenotypes as in physical appearance. My skin tone is very different from yours, okay, your skin tone is very different from your friends, everywhere like that. Why? Because over here you have polygyny. Multiple genes located on multiple chromosomes, my dear children, probably might be coding for what? A character that is your skin color, and the same goes with your height as well. So multiple genes are responsible for it. Suppose over here I have capital A and then capital A. Over here I have capital B, capital B. Over here I have capital C gene. All of them are responsible for giving me the, what do you call, the darkest skin tone, which is capital A denoted by capital A, capital A, capital B, capital B, capital C, capital C. Now suppose over here the allele is changed to small a. The skin color will reduce. It will be something like this. The genotype is like this. The phenotype is also going to be changed. Now suppose it's small b, again it will be changed. Small c, again it will be changed. You get it? So multiple genes are responsible for one particular character, which is why we would have different combination of it, which is why we also show different phenotypes of it. Okay. Now accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why is it happening? Because the enzyme, the gene that codes for phenylalanine hydroxylase is being mutated. That is why it is getting accumulated. So how do we explain all of this? Hemophilia, non-stop bleeding occurs when a single protein that is a part of cascade of proteins involved in the clotting of blood is affected. Turner syndrome, females are usually sterile because they have only one X chromosome, leading to underdeveloped ovaries and lack of gamete production. Morgan's dihybrid cross, yellow body and white eye genes in Drosophila show less recombination. Why? Because they are very located very close to each other on the same chromosome, exhibiting what? Genetic linkage. They are tightly linked. You have to use that term. It's not used over here, but you need to use that. Tightly linked. Now skin color in humans, it shows different phenotype due to polygenic inheritance where multiple genes contribute to the phenotype, leading to the range of skin color variation. That is why we have so many variations. The lightest skin color is small b, small b, small a, small a, small b, small b, and small c, small c. Okay, all of them are chotu. Now phenylketonuria, phenylalanine accumulates in the body due to defect in the enzyme phenylalanine hydroxylase, which converts phenylalanine to tyrosine, causing what? The toxic buildup that you see. We explained it, right? Yes. Now going ahead to the next question, define aneuploidy. Give two examples of aneuploidy. What is aneuploidy? Whenever you have an addition or a subtraction or reduction or addition of a chromosome, this condition is where you call as aneuploidy. Give two examples of aneuploidy, you know that. One can be Down syndrome, another can be what? Turner syndrome, either way. And they've also asked, mention the karyotype of Klinefelter syndrome of affected individual and write its symptoms. So let's see all of it together here itself. So aneuploidy is a condition when an organism has an abnormal number of chromosomes, either due to gain or loss of one more chromosomes. It is caused by non-disjunction during the cell's division. Very, very important. What do you mean by that? So usually when the cell divides, my dear children, what happens? The chromosome there divides and then the cell divides. But probably the chromosomes might not be dividing properly. They might be still together. The chromosomes which has to go to either sides, they are not going. So non-disjunction is happening, my dear children, during cell division, which is why there can be a gain or probably a loss. So one that who takes it, takes all of it. Here and here it will be less. So here it will be loss, in one gamete it will be loss, in another gamete there will be a gain of it. So this is aneuploidy, a condition. This is example of how chromosomal disorders are being formed. So there is a condition like aneuploidy and there is condition like polyploidy. Polyploidy is usually seen in the case of plants, my dear children, where what is happening, you are just not talking about one chromosome. There the whole set of the chromosome can be going to one. Get it? There might be a problem with the whole set of chromosome. That happens in the case of polyploidy. Here in the case of aneuploidy, you are talking about one or two things, that is either one chromosome or maybe two chromosomes, that's it. In which over here an abnormal number of chromosomes are there due to gain or loss of one or more chromosome. This is due to what? Non-disjunction during cell division. Example, Down syndrome, trisomy of what? 21st chromosome. So in total they would have how many number of chromosomes? 47 chromosomes. Monosomy, in Turner syndrome there is one chromosome which is being lost. It's not there, right? So instead of having 46 chromosome, they would be having 45 chromosome. Now the karyotype of affected individual of Klinefelter syndrome. My dear children, you tell me, how many autosomes will be there in Klinefelters? 44 of them. It's safe. Now we talk about the sex chromosome. It is XXY condition. It is what? XXY. So I can write it like this, 44A That means I am denoting the number of autosomes and then I am telling about the sex chromosome. One is extra. So in total 44, 45, 46, 47 chromosomes are there. Or else I can just write like this, 47 XXY. So I get to understand that this is the one that is extra. So I can write like this where I specify about autosomes, or I can directly write like this also. This is the karyotype. Ma'am, what is the karyotype of Turner syndrome? Again over here 44 autosomes are there and then you have XO condition. You have what? XO condition. So you can write like this, 45 XO. Either you write like this or you write like this. Okay. Now, symptoms of Klinefelter syndrome, very important. So this had to be a male, but obviously there are female characteristics associated with it. Why? Because you have an extra X chromosome. So underdeveloped secondary sexual characteristics will be there. They would not have all the male secondary sexual characteristics. They will be very underdeveloped. Gynecomastia is there, that is enlarged breast in the case of males. They are males but they are showing female character. This is known as gynecomastia. Infertility. Why? Because lack of spermatogenesis is happening over here. X chromosome is there to dominate over things, right? And tall stature with long arms and legs. Learning difficulty in some cases, you don't need to write that. At least you write these 3 points. That would be enough. Okay, underdeveloped secondary sexual characteristics, gynecomastia, that is enlarged breast in the case of males, and infertility due to lack of what? Spermatogenesis, that is happening over here. Perfect. They have underdeveloped secondary sexual characteristics, which is why they would not be showing spermatogenesis, my dear children. I hope that's also clear. Okay, fine. Now, what is incomplete dominance? Explained with reference to flower color in snapdragon. What do you mean by incomplete dominance? So, we know, for example, when you talk about capital R, or when you say small r, we know that capital R means probably this is R is something dominant. And over here I am writing R is for red and the small r is for white color. So, my dear children, when you form the gametes over here, okay, this is the parent, this is the gamete and this is the F1. Write it properly, parents, gamete and all. Okay, I am not writing it over here. So this and this fuses, you get capital R smaller, this and this fuses, you get capital R smaller, this and this again capital R smaller, this and this capital R smaller. All of where you have capital R, so you are expecting it to what? It to be red in color, but the twist in the story is that all of them are pink in color. That means what? This is not completely dominant over this. This is an example of incomplete dominance. They are not completely dominant over each other. Okay, now ma'am, I want to see the whole cross. What happens in the whole cross? Now Mendel was the person who did it got confused, so he wanted to cross it again. So when he crossed it, my dear children, what happened? Let's see. So capital R, small r in the gametes, again capital R, small r in the gametes. This and this goes, you get capital R. This and this goes, you get capital R, small r. This and this, capital R, small r, and this and this, small r, small r. So if you see, there is, both of them are capital R, so this will be red. This would be not completely dominant, so this would be pink. and over here this has just smaller, so this would be white. So if we talk about the phenotypic ratio, how do they look? 1 red, 2 pink and 1 white. 1 red, 2 pink and 1 white. Now just a second, okay, just give me a second, I'm getting a call. Okay, so this is the phenotypic ratio. Now what is the genotypic ratio, my dear children? Capital R, 1. Capital R, small r, 2 of them. Small r, 1 of them. So again, genotypic and phenotypic ratio remains the same. And this is what you mean by incomplete dominance. Over here you can see incomplete dominance. One is not completely dominant over the other, which is why in the next generation also you can see the same thing happening. Okay, and snapdragon is also known as antirrhinum. Do not forget it. Antirrhinum, that is what is known as. So a phenomenon where both the alleles of a character express incompletely, producing a new intermediate phenotype in the heterozygous, only in the heterozygous condition. When it is like this, that's when it produces, okay. This is known as incomplete dominance. Coherence crossed homozygous red flower with white flower plant, and in F1 generation all hybrids were produced which were pink in color. The dominant allele capital R was not completely dominant over the recessive allele small r, and this made it possible to distinguish capital R small r as pink from capital R red and small r white. When F1 pink flowering plants were self-crossed, the F2 generation produces 25% red flower, 50% pink flower and 25% red flower. This is what is exactly explained over here. 25%, 25% white, 25% red and 50%. 2 of them are pink in color. So you have to explain in the same manner. Do not write too much of things, okay, write in points, make it short. And my dear children, what do you have as of now with you guys? You have AI. So what you need to do is, whenever you're not able to figure out how you need to answer, is that put the question out there and tell them to explain the best possible way for your PU2 examination in points. You have to mention all of it in points, in short points. This is a 5-marker question and things like that. They will give you perfect answer out there, okay, if at all you face trouble. Now comes to the last question of today's session. With that, I think we can end it. And before going ahead, let me tell you, if at all you liked this session, okay, if at all you thought that, okay, ma'am explained well, or you understood things well, put it down in the comment section. I will be really happy to read it. And if at all you have doubts around any question out there, put it down there. I will be here to explain you out, even in the comment section. Okay, done? So write the schematic representation of one gene inheritance and Pisum sativum. What is Pisum sativum? It is what? The garden pea out there. It has 7 characteristics. I hope you know 14 traits. Okay, I made you remember that in your previous session. Now, schematic representation of one gene inheritance. How do you show? One gene means you're talking about a gene probably that is can be height or anything. So I am taking the gene for height. So over here I have capital T, I have small t. This is homozygous parent, homo as in same, homozygous parent, homozygous tall, homozygous dwarf. Okay, this is parent, the gametes will be capital T, small t over here. This and this crosses, I get capital T, small t, this and this, capital T, small t, this and this, capital T, small t, this and this, capital T, small t. All of them are tall in nature. Why? Because you have capital T over here. This is what F1 generation. Now you do F2 generation where you cross this and this, my dear children. So over here capital T small t, then capital T small t. So when you cross this, you get one capital T, one capital T small t, again one capital T small t, again small t. So one of them is going to be tall, one of them is going to be dwarf, 2 of them are going to be no, 3 of them are going to be tall. Why? Because here it is completely dominant. So 3:1 ratio is what you get. This is about the phenotypic ratio. If you talk about the genotypic ratio, 1 capital T, 1 small t, 2 of them capital T, small t. So it will be 1:2:1. This is the genotypic ratio. So you have to mention properly line by line. Do not make it like this, make it like this, okay? And sure, over here the perfect example is given for you so that you do not refer to Jasmine ma'am but to this particular image. Got it? Will you be able to do it? Always remember genotypic ratio and phenotypic ratio has to be mentioned, and whenever you mention that as given over here, you have to tell genotypic ratio is what? 1:2:1. What do you mean by that? 1 pure tall, 2 hybrid tall, 1 pure dwarf. Okay, and you are mentioning small t, capital T and capital T, sorry, capital T, small t and capital T here as well. Similarly, when you are saying 3:1 is the ratio, what is 3? 3 is tall, 1 is what? Dwarf. Mention that. Okay, and with that, we end today's session. I hope it was helpful. So I tried to explain you as many things as possible out there, and if at all you have some doubts, as I said, please put it down in the comment section, my dear children. And if at all you think that this session was super helpful for you, share it with the friends who are in need. And with this, we have completed Principles of Inheritance and Variation, and I can guarantee you 13 on 13 marks in your examination if all the questions are coming from here only. Okay, so that's all. Wishing you all the best. Bye bye. Take care.